You can start, beta. So, good morning to all of you. Good morning to all of you. Yesterday we were doing the Zwitter ion, so we have to continue with that. And I was telling you the difference between the Zwitter ion and the normal ion. So Zwitter ion carries both the charges of the positive charge and the negative charge. Both the charges are carried by the Zwitter ion, and that is why we call it a hermaphrodite species. That means when we see a Zwitter ion, it looks like this, which has a positive charge connected to C, which is connected. If we make glycine, the variable group will be H, and we have COO with a negative charge. There is an internal transfer of hydrogen from carboxylic acid to the amino group. Internal transfer of oxygen from the carboxylic acid to the amino group. So this amino group gets a positive charge and becomes H3N, whereas this gets a negative charge and becomes COO. So this is a Zwitter ion. It is a hermaphrodite species and is Zwitter ion, which is seen in case of amino acids. The amino acids, they form Zwitter ion mostly in soluble form. Mostly in soluble form, this forms the Zwitter ion. Just a minute. Let me admit few students. Yes. So this is what happens. Now, I told you what happens to the Zwitter ion under how do the Zwitter ion respond? Response of Zwitter ion under different pH. Response of Zwitter ion under different pH. So when the pH is something and the pH is acidic or the pH is alkaline, what happens to the Zwitter ion? So first we will take when the pH is alkaline or when an alkali is added to it. So first we take when an alkali is added to it. That means when we add OH, when OH is added to it. So let's make the Zwitter ion. We will make the Zwitter ion first. So it is H3N with a positive charge C. This is glycine. Zwitter ion formed by glycine. So I have made it like this because the variable group is H. So it is glycine. Or we can simply write R also. But here I have made it glycine. So I have done it like this. So now what happens to this? Or it is not, we can write this H here also. So it can be like this also. We can write it this way also. So to this, when an alkali is added, what happens to the Zwitter ion? This I'm going to show. This positive has an interaction with this negative has an interaction with the positive. So what are we going to get? This will lose its charge. CH, H, the charge will remain there. This has lost the charge, and H of this combines with OH to form H2O. This is the reaction which takes place. So only negative charge is left on the Zwitteron when an alkali is added. So what do we get? We get a glycinium anion. Glycinium anion. This is what we get. With the formation of water. With the formation of water, we get glycinium anion. So is it clear to you? Yes, beta, is it clear to you? This reaction is clear to you? Okay, I'm getting the response. Okay, fine. Now I'll show you what happens when acid is added to the glycinium ion, to the Zwitter ion. What happens to the Zwitter ion then? So the second thing which I'm going to tell you now, when an acid is added to it, it means the Zwitter ion. When an acid is added to it, and what is that? H positive. Now here, The reaction is H3N. This is deuterion. CH, H, COO negative. This is deuterion. So to this, when an acid is added, this plus has an interaction for the positive, has an interaction for negative. So what is going to happen? The positive has an interaction for negative. So what is going to happen? This will remain the same with the positive charge. Here CH, H here, and here this will become COOH without any charge. So this has only positive charge, and this is glycinate. Glycinate cation. This becomes glycinate cation. So this is what happens when an acid is added to the deuterion. So when an alkali is added to the deuterion and when an acid is added to the deuterion, this is what happens. So anything in the amino acids which you could not understand or you want me to repeat? Yes, beta. Anything in deuterion which you want me to repeat? Okay, Deepika. Others? Find other. Yes, Yashika, Amrita, Harsha, Jasleen, Siksha, Lalit. Okay, now I start with the next, and that is lipids. So I start the next, which is lipids. What are lipids basically? They are inorganic or organic? They are organic compounds. Okay. If we see, they are composed of two things, fatty acids. They are composed of two things, fatty acid plus alcohol. They are composed of fatty acids and alcohol. Another point, their characteristic feature is they are insoluble in water. Another characteristic feature is they are soluble in organic solvents. So this is their characteristic feature. They are organic compounds. They are composed of fatty acids and alcohols. They are insoluble in water. They are soluble in organic solvents. Now, if we say chemically, their chemical structures, they are long chain fatty hydrocarbons. They are long chain hydrocarbons. That means composed of carbon and hydrogen, and they have terminal carboxylic group or terminal carboxylic acid. So how we are going to define this? This is a class of, how we will define this? We are going to define it like this. Class of organic compounds, lipids are these are class of organic compounds which are composed of fatty acids and alcohol, or are derivatives of fatty acids, which are soluble I write, and are soluble in in organic solvents and are soluble in organic solvents but are insoluble in water. If we see chemically what they are, chemically, basically they are composed of lipids, are long chain hydrocarbons with terminal carboxylic groups. With terminal carboxylic groups. So if we see, they are long chain hydrocarbons. Long chain hydrocarbon, we have hydrogen here, long chain hydrocarbons with terminal carboxylic group. So I have drawn a lipid long chain hydrocarbon with terminal carboxylic group. Is it clear? This is something about the introduction of lipids. Is it clear to you all? Yes? Anything you want to ask me? Terminal carboxylic group is present, the terminal carboxylic group, and these are long chain hydrocarbons. Is it clear, beta? Yes? I'm not getting any response? Fine, fine. So now I come to the types. The lipids are of three types. Three types of lipids. There are three types of lipids. Their names are the first one is simple lipid. The second one is complex or conjugated, anything we can see. Conjugated or complex lipids. I'm writing just L. And the third one is derived lipids. So we have three types of lipids, the simple lipids, conjugated lipids, and derived lipids. Now, what are these simple lipids? Simple lipids, they are composed of, or they consist of fatty acids plus alcohol. They consist of fatty acid and alcohol. Their examples are true fats and waxes. In true fats, the alcohol is glycerol. Okay, true fats have glycerol. The alcohol group is glycerol, whereas the waxes have other alcohols, but not glycerol. Other alcohols are present in waxes, but not glycerol. So these are true fats. When glycerol is present, fatty acid plus glycerol, they form true fats. When fatty acid plus other alcohols other than glycerol is present, we say that these are waxes. Now the second type. In the second type, which is conjugated, I'll draw a line so that you understand it better. We have fatty acids plus alcohol plus one additional group. So when the lipid has fatty acid plus alcohol plus any additional group, we call it conjugated or complex lipid. This is known as conjugated or complex lipids. The example is phospholipid. One such example is phospholipid. This is present in the plasma membrane. Some more examples of this is if protein, here the phosphorus is the additional group. Phosphorus is the additional group in this. The lipid has fatty acid. In phospholipid, it has fatty acid, alcohol plus phosphorus. So this becomes a conjugated or complex lipid. The second example of this is lipoprotein. Lipoprotein. Here, lipoprotein means what it has. This is a lipid, that means it will have fatty acid plus alcohol, and the additional group is a protein. So we call it lipoprotein. The third example is glycoprotein. Glycolipids, not protein. Glycolipids, here the glycolipid consists of fatty acid plus alcohol, and the additional group in this case is a carbohydrate. Glyco is a carbohydrate. So this is another type, the conjugated or the complex protein lipids. We come to the next type, which is derived lipids. Derived lipids are the lipids which on hydrolysis gives you simple lipids as well as conjugated lipids. That means on hydrolysis. On hydrolysis means, lysis always means breakdown. Hydrolysis, breakdown in the presence of water. So when hydrolysis takes place, when the breakdown takes place on hydrolysis, they yield simple and conjugated lipids. On hydrolysis, they yield simple and conjugated lipids, and these are known as derived lipids. The examples of these are sterols. Sterols, example of any sterol, cholesterol. Then some terpenes are also there, which are in the derived lipids. So we have three types of lipids: the simple lipids, the conjugated lipids, and the derived lipids. So when fatty acid and alcohol only is present, we call it simple lipids. When fatty acid, alcohol, and an additional group is present, we call it conjugated or complex. And when we have such a lipid which on hydrolysis gives you simple and conjugated both type of lipids, then we call it a derived lipids, and sterols are those. That means cholesterol is a type of derived lipid. When cholesterol is broken down or its hydrolysis takes place, it gives you simple lipids as well as conjugated lipids. Is it clear? Is it clear, beta? Everything is visible to you? Any doubt, any query in this? Okay. Any question? Any question, any query in this? Okay, Amrita. Yes, Yashika, Harsha. Okay. Terpenes are those lipids which on hydrolysis yields you simple and conjugated. It is a combination of the simple and the conjugated lipids. Okay. Fine. So I come to another point, that is about the fatty acid, and I'll tell you the types of fatty acid. So because fatty acid is an important component of lipids. Fatty acids are important components of lipids. So I come to the next point. Fatty acids are of two types. Fatty acids are of two types. Can anybody tell me what are the two types of fatty acids? You have done it before also. Yes, beta? Yes? Anybody can tell me the two types of fatty acids? Yes, bache? Yes? Okay, fine. The two types of fatty acids are saturated fatty acids and unsaturated fatty acids. Saturated fatty acids and unsaturated fatty acids. The difference between the two. What are saturated fatty acids and what are unsaturated? Okay. The saturated fatty acids, these are solids at room temperature. So if you check the at room temperature, these saturated fatty acids, these are solids at room temperature. These are solids at room temperature and these are liquids at room temperature. I'm just writing liquids. These are solids at room temperature. These are liquids at room temperature. Now, if we see these which are solids at room temperature, obviously their melting point will be high. So they have high melting points. And they have low melting point. I'm just writing low MP. Okay. Now, if we see in this, the saturated, no double bonds are present. No double bonds. No double bonds are seen in case of saturated, whereas double bonds are present in unsaturated. Double bonds are present. Now, these saturated are mostly found in animal tissues. These are mostly found in animal tissues, and if we see this, these are mostly found in plant tissues. You know what is vegan diet? Have you heard of vegan? Yes, vegan.
Yes.
What is vegan? What is that?
They don't eat. They don't have any milk products, and neither the meat of the animals.
Yeah. See, when they don't take meat, that is non-vegetarian. But they know animal products, basically milk and milk product. It is
They
not
don't have the
animal
milk
product.
products.
Milk and milk products.
The milk.
So
Yes.
most what happens, the animal products like milk and milk products, they increase the cholesterol in the blood. So the people who suffer from a high cholesterol level, they usually become this. You know that these milk products, they increase the cholesterol level. So mostly they become vegan. Now you think they do, that means they do not eat any sort of paneer or milk? Do they have some other source of milk or paneer? Yes, do they have? Yes, they take soy milk, soy milk and the tofu is the cheese which is made from the soy milk. So they consume that. Now, if we see, this is found in animal tissues, this is found in plant tissues. If we see the general formula of this, the general formula of the saturated fat, general formula I'll take for both. This is CnH2nO2. This is the general formula for this, and the general formula for this is CnH2n minus 2xO2. This is the general formula for the unsaturated fat. The example of saturated fat. Example is palmitic acid. The spellings are of palmitic acid, PALMITIC acid. And the formula of palmitic acid is C16H32O2. And the example of this is oleic acid, OLEIC, oleic acid. And the formula for this is C18H34O2. So this is about the saturated and unsaturated fatty acids. Is it clear, beta? Saturated and unsaturated fatty acids is clear? Yes. Now we come to another point. Now we will study about, I can teach you the lipids in detail also, but we have to see what we have in our syllabus. That is why I'm teaching you only this much. But if you want, I can teach you the lipids in detail. I'll see about it. Okay, so let's start with the bonds in lipids. Bonds in lipids. The bond which is found in lipid is ester bond. Ester bond is seen in lipids. And you know what are esters? What are esters? In chemistry, you must have studied esters. What are esters? Yes, beta, what are esters? You must have studied in chemistry esters. Anybody, if you know esters? These are the compounds in which one OH of those compounds, these are replaced by alkyl group, in which OH is replaced by alkyl group. Okay, these are esters. So ester bonds are present in the lipids. Now what happens? We have, on the basis of number of fatty acids, number of fatty acids present we have monoglycerides, diglycerides, and we have triglycerides. We have mono, we have di, and we have triglycerides. Now, what are these mono, di, and triglycerides? This I'm going to explain now. So here, if we have one alcohol with one fatty acid, it is a monoglyceride. If we have one alcohol plus two fatty acids, it is a diglyceride. If we have one alcohol plus three fatty acids, it is a triglyceride. So we have mono, we have di, and we have triglycerides. When one, two, or three fatty acids are present, we call it when one mono, when two di, and when three triglyceride. Now, how the bond formation takes place? This is what I'm going to explain now. Suppose we have this. Now, I'll explain you bond formation. We have this alcohol CH2OH, CHOH, CH2OH. We have suppose this alcohol, and this combines with a triglyceride HOOC and R1. HOOC R2, HOOC R3. This is a triglyceride. This is a triglyceride. Now, what happens when it combines? When the bond formation takes place here, this here, this formation, this combination takes place, and as a result, what do we get is here CH2, CH2, OOC, R1, CH, OOC, R2, CH2, OOC, R3. This is what we get along with the formation of water. These are 3 fatty acids along with the formation of 3H2O. This is a triglyceride. A triglyceride is formed this way. When 3 fatty acids, they join with one alcohol. This is one alcohol, and these are 1, 2, 3 fatty acids. So when one alcohol combines with 3 fatty acids, we get triglyceride with the release of 3 molecules of water. Is this reaction clear to you? The formation of the ester bond? The formation of ester bond, beta, is it clear? Yes, beta. Formation of ester bond is clear. Yes, bache? Fine. The same thing you can show by when 2 fatty acids combine, how the diglycerides are formed. So alcohol will remain 1, CH2OH, CHOH, CH2OH. This is the alcohol which is combining with 2 fatty acids. So I'm writing HOOCR1, HOOCR2. 2 fatty acids are combining, and when the combination takes place, because 2 fatty acids are combining here, we will get a diglyceride. And how does that diglyceride look like? So it will be CH2OOCR1 here, and here we'll write just CHOH, and at the third place we write CH2OOCR2. This is a diglyceride which is formed. This is a diglyceride which is formed. So this same way you can make the monoglyceride also, and the formation of triglyceride, diglyceride is by ester bond formation. In case of lipids. In case of lipids, it is ester bond formation. Is it clear to you, beta? Any query in this? Yes, bache, any question in this? Any query? Okay. Any question to anybody? Yes, Yashika, Amrita, Deepika, Siksha, Jasleen, Zainab. Zainab is very quiet. Yes, okay. Zainab. Fine. So here, this, fine, Jasleen. Here I finish with the lipid portion, and we'll continue with the next one. or I may give you some detail of this. Fine. So the time is over. I end the meeting. Okay, stay safe, children. Good day.